Hi all, welcome to the lecture about electroencephalogram. This topic is from Module 4 of EE372 Biomedical Instrumentation. Myself, Renu George, Assistant Professor, Department of Electrical and Electronics Engineering, HARSET. Electroencephalogram, commonly known as EEG, is a continuous recording of electrical activity of the brain. During an EEG test, small cup or disc type electrodes are placed on the scalp. They pick up brain's electrical signals and send them to a machine called electroencephalograph. It records the signals as wavy lines onto a computer screen or paper in order of microvolts. Experiments have shown that the frequency of EEG seems to be affected by mental activity of a person. For understanding this, let's consider the waveforms associated with different stages of sleep. An awake or alert person usually displays an unsynchronized high frequency EEG. A drowsy person, particularly one whose eyes are closed, often produces a large amount of rhythmic activity in the range 8 to 13 Hz. In light sleep, a large amplitude, low frequency waveform emerges. Deeper sleep generally results in even slower and higher amplitude waves. EEG frequency bands or rhythms can be arbitrarily classified into four. First one, delta waveforms having frequency below 3.5 Hz. Second one, theta waveforms having frequency ranging from 3.5 Hz to 8 Hz. Third one, alpha waveforms with frequency ranging from 8 Hz to 13 Hz. Fourth one, beta waveforms having frequency above 13 Hz. Most humans seems to develop EEG patterns in the alpha range when they are relaxed with their eyes closed. This condition seems to represent natural frequency of the brain. As soon as the person becomes alert or begins thinking, the alpha rhythm disappears and is replaced with desynchronized pattern, generally in the beta range. Before moving to the details of EEG machine, let's look into the anatomy of nervous system. As you all know, nervous system controls and coordinates all essential functions of human body. The human nervous system can be divided into two, the central nervous system and the peripheral nervous system. Central nervous system includes brain and spinal cord. It is the site of information processing and control. Peripheral nervous system comprises of all nerves of body associated with central nervous system. Let's look into the details of central nervous system. Main parts of central nervous system are brain and spinal cord. The brain is a collection of cell bodies and fibers located inside the skull, where it is protected from light as well as from physical, chemical or temperature shock. At its lower end, brain connects with spinal cord, which also consists of many cell bodies and fiber bundles. The central nervous system is considered to be bilaterally symmetrical. A very general look at the anatomy of brain should be helpful in understanding the functions of nervous system. Figure shows a cutaway showing some of major structures of brain. The part of brain that connects to spinal cord and extends up to the center of brain is called brainstem. The essential parts of brainstem are medulla, sometimes called medulla oblongata, which is the lowest section of brainstem itself. The pons located just above medulla and protruding somewhat in front of brainstem, and the upper part of brainstem called the midbrain. Medulla seems to be associated with control of some basic functions responsible for life such as breathing, heart rate, kidney functions, etc. For this purpose, medulla seems to contain a large number of timing mechanisms, as well as important neuronal connections. The pons is primarily an interconnecting area, play a vital role in salivation, feeding, facial expression, etc. In addition, the pons contain relays for the auditory system, spinal motor neurons, etc. Behind the brain is the cerebellum. The cerebellum plays a vital role in man's ability to maintain his balance. Almost completely surrounding the midbrain, thalamus and hypothalamus are the structures of cerebrum. The cerebrum forms the major part of human brain. The outer surface of cerebrum is called cerebral cortex, which contains 9 billion to 12 billion neurons found in human brain. The cortex is actually a thin layer of neurons at the periphery of brain, which contains folds to provide a greater amount of surface area. About different lobes of brain. Each side of brain contains 4 lobes. The frontal lobe is important for cognitive functions and control of voluntary movement or activity. The parietal lobe processes information about temperature, taste, touch and movement, while occipital lobe is primarily responsible for vision. The temporal lobe processes memories, integrating them with sensations of taste, sound, sight and touch. Second part of nervous system is peripheral nervous system. Peripheral nervous system can be divided into two: somatic nervous system and autonomic nervous system. Somatic nervous system transmits impulses from central nervous system to skeletal muscles. Autonomic nervous system transmits impulses from central nervous system to involuntary organs and smooth muscles of the body. Autonomic nervous system can be further classified into sympathetic nervous system and parasympathetic nervous system. The sympathetic nervous system tends to mobilize the body for emergencies, whereas the parasympathetic nervous system tends to conserve and store bodily resources. Nervous system is made of cells called neurons or nerve cells. Let's look into the structure of a neuron. A neuron is a microscopic structure composed of three major parts, namely cell body or soma, dendrites, and axon. Axon branches near its ending divides into two or more terminals. The portion of axon immediately adjacent to the cell body is called axon hillock. The branches that leave main axon are often called collaterals. Certain types of neurons have axons or dendrites coated with a fatty insulating substance called myelin. The coating is called myelin sheath and the fiber is said to be myelinated. In some cases, the myelin sheath is interrupted at regular intervals by the nodes of Ranvier, which improves speed of transmission along the nerves. Outside of central nervous system, the myelin sheath is surrounded by another insulating layer called neurilemma. This layer is thinner than myelin sheath and continuous over the nodes of Ranvier. Is made up of thin cells called Schwann cells. Both axons and dendrites are called nerve fibers, and a bundle of individual nerve fibers is called a nerve. Nerves that carry sensory information from various parts of body to brain are called sensory nerves or afferent nerves. Whereas those carry signals from brain to various muscles are called motor nerves or afferent nerves. Cell bodies and small fibers in brain are grey in color and are called grey matter. Whereas the myelin coating of larger fibers has a white appearance, so that a collection of these fibers is referred to as white matter. Collections of neuronal cell bodies within the central nervous system are called nuclei, whereas similar collections outside central nervous system are called ganglia. Neuronal communication. Neurons are capable of being excited. When excited, neurons generate action potentials. In neurons, these action potentials are of very short duration and are called neuronal spikes or spike discharges. Information is usually transmitted in the form of spike discharge patterns. The spike discharge pattern from a single cell neuron in red nucleus of cat as shown in the slide. Synapse. A nerve impulse is transmitted from one neuron to other through junctions called synapses. The membranes of pre and post synaptic neurons are separated by a fluid-filled space called synaptic cleft. The axon of one neuron can activate the dendrite of cell body of another by producing a chemical that simulates the membrane of a dendrite or cell body. Arrival of an action potential at an axon terminal releases a chemical, acetylcholine, which excites the adjacent membrane of the receiving neuron. Because of the close proximity of transmitting axon terminal to the receiving membrane, the time of transmission is still quite short. The possibility that some of the chemical may still be present after the refractory period is eliminated by the presence of acetylcholine esterase, another chemical that breaks down acetylchlorine as soon as it is produced, but not before it has been able to initiate its intended action potential in the nearby membrane. There are really two kinds of communication across a synapse, excitatory and inhibitory. Several axons from different neurons are in communication with the input of any given neuron. Some act to excite the membrane of the receiver, while other tends to prevent it from being excited. Graded potentials are variations around the average value of resting potential. When this graded potential reaches a certain threshold, the neuron fires and an action potential develops. The excitatory gradient potential is called excitatory postsynaptic potential, and an inhibitory graded potential is called an inhibitory postsynaptic potential. Electrodes and microelectrodes are used in the measurement of gross and single neuronal firings. When the measurement is made between a single electrode and a distant indifferent electrode, the measurement is defined as unipolar. When the measurement is obtained between two electrodes spaced close together along a single axon or a nerve, the measurement is called bipolar. Several methods of measuring neuronal spikes have been developed. One is gross nerve firing measurement, which is obtained when a relatively large electrode is placed in the vicinity of a nerve or a large number of neurons. The result is the summation of the action potentials from all the neurons in the vicinity of the electrode. The gross measurement of a multiple unit neuronal discharge is shown in the slide. Full width covers time span of 500 milliseconds. Maximum peak-to-peak amplitude is approximately 145 microvolts. For extracellular measurement, a microelectrode is located just outside the cell membrane. Extracellular measurement of a unit discharge from red nucleus of a cat is shown in figure. Peak-to-peak height is approximately 180 microvolts. For intracellular measurement, a microelectrode is penetrating the cell. Intracellular measurement from abducens nucleus of a cat is shown in the slide. Spike height is about 61 millivolts and each horizontal division is 0.5 millisecond. Electroencephalograph or EEG machine. Electroencephalograph is an instrument for recording the electrical activity of the brain by placing surface electrodes on the scalp. Monitoring the electroencephalogram has proven to be an effective method for diagnosing many neurological illness and diseases. About electrodes used in electroencephalograph. Several types of electrodes are used to record EEG. These include peel and stick electrodes, silver-plated cup electrodes, and needle electrodes. EEG electrodes are smaller in size than ECG electrodes. They may be applied separately to the scalp or may be mounted in special bands which can be placed on the patient's head. If electrodes are intended to be used under the skin of the scalp, needle electrodes are used. They offer the advantage of reducing movement artifacts. A suitable electrolyte paste or jelly is used in conjunction with the electrodes to improve the electrical contact. EEG electrodes give high skin contact impedance as compared to ECG electrodes. EEG preamplifiers are generally designed to have a very high value of input impedance to take care of high electrode impedance. Placement of electrodes on the scalp is commonly dictated by the requirements of measurement to be made. A pattern of electrodes on the head and the channels they are connected to is called a montage. Montages are always symmetrical. The reference electrode is generally placed in a non-active site such as forehead or earlobe. EEG electrodes are arranged on the scalp according to a standard known as 10-20 system adopted by American EEG Society. The system is so named because electrode spacing is based on intervals of 10 and 20% of distance between specified points on the scalp. The 10-20 electrode configuration is illustrated in the figure. Electrodes can be identified according to their position on the head. FP stands for frontal polar side, F for frontal side, C for central position, P for parietal position, T for temporal position, and O for occipital position. Odd numbers refer to electrodes on the left side of head and even numbers represent those on the right, while Z denotes midline electrodes. The basic block diagram of an EEG machine is shown in figure. An electron encephalogram is recorded simultaneously from an array of many electrodes. The electrodes are connected to separate amplifiers and writing systems. Commercial EEG machines have up to 32 channels. Now moving on to the functions of each block. First one, electrode montage selector. EEG signals are transmitted from electrodes to the jack box and then to montage selector. The montage selector on analog EEG machine is a large panel containing switches that allow the user to select which electrode pair will have signals subtracted from each other to create an array of channels of output called montage. Each channel is created in the form of input from one electrode minus the input from a second electrode. Montages may be bipolar or unipolar. Bipolar montages are made by subtraction of signals from adjacent electrode pairs. Unipolar or referential montages are made by subtracting the potential of a common reference electrode from each electrode on the head. Next, about amplifiers. Every channel has an individual multi-stage AC coupled very sensitive amplifier with differential input and adjustable gain in a wide range. The preamplifiers used in electroencephalographs must have high gain and low noise characteristics because the EEG potentials are small in amplitude. In addition, the amplifiers must have very high common mode rejection to minimize stray interference signals from power lines and other electrical equipments. The amplifier must be free from drift so as to prevent the slow movement of recording pen from its center position as changes in temperature, etc. About filters. Filters are used for selecting frequency response. The frequency range of standard EEG machine is from 0.1 Hz to 70 Hz. High-pass filters are employed to control upper cutoff frequency. The values that can be selected are 15, 30, 70 and 300 Hz. Low-pass filters are used for removing muscle artifacts due to contraction of scalp and neck muscles. Several values selected are 5.3, 1.6, 0.53 and 0.16 Hz. Notch filters are employed to eliminate mains frequency interference. They are sharply tuned at 50 Hz. About writer unit. Writer unit consists of writing part and paper drive. The writing part of an EEG machine is usually an ink-type direct writing recorder. Paper drive is provided by synchronous motor. An accurate and stable paper drive mechanism is necessary and it is normal practice to have several paper speeds available for selection. Speeds are 15, 30 and 60 mm per second. A timescale is usually registered on the record by one or two time marker pens which make a mark once per second. Timing pulses are preferably generated independently of the paper drive mechanism in order to avoid difference in timing markings due to changes in paper speed. The output from amplifier can also be displayed in oscilloscope or a computer by employing an analog to digital converter. Modern EEG based machines are mostly PC based. The system can store up to 40 hours of EEG. The EEG is displayed on a color monitor. For further reading, refer textbooks Biomedical Instrumentation and Measurements by Cromwell and Handbook of Biomedical Instrumentation by R.S. Kanpur. Thank you.